Welcome to my channel Adhyan with Flow. So let's start with our other cell organelles, cytoskeleton. Cytoskeleton mainly used by motility, locomotion, movement. Mainly, I'm saying mainly. There is three types of cytoskeleton in our body: microtubule, microfilament, and intermediate filament. There are three types of cytoskeleton. First is microtubule, second is microfilament, and third is interfilament. They both useful in motility. They both are useful in motility. They mainly participate in cell division time, other motile locomotion movement. Alright or not? Where they located? They located in cytoplasm. They also attach with, this is the plasma membrane, they are also attached with plasma membrane. What they do? The function of skeleton is, look at now, skeleton, they give rigidity, they give support, they give strength, they give motility, they give maintenance of shape of the cell. They maintain what? Shape of the cells. Look at here, guys, the structure. If you want to see, look at their arrangement. This is the 9+2 arrangement. Count 1, 2, 3, 4, 5, 6, 7, 8, 9. And plus 2, this is U1 and 2. What is this? This is the plasma membrane. These are microtubules. If we saw the top view of, then microtubules look like this. But this arranged in this manner. Look at here, guys. Now you can better understand. Okay. What are these? Connected interdoublet bridge. The connection between tubules, microtubules. These are center microtubules and these are radial spoke. And what is this? This is the sheet. This structure is important, guys. This structure is important. Alright. Now, cilia and flagella. Cilia. They present in the outer surface of organisms. And flagella also present at the anterior part of the organ surface. Cilia and flagella, they are hair-like outgrowth of the cell membrane. Look at here, guys. This is the cell membrane and these are the outgrowth. Cilia are huge in number and flagella are less in number. Cilia are small structures which work like oars, causing the movement, guys, movement of either the cell or surrounding fluid. They provide movement to this organism. Flagella means cilia are small and maximum in number, and flagella are long and less in number. So flagella are comparatively longer and responsible for cell movement. This is also helpful in movement. Flagella provide also movement in organism. The prokaryotic bacteria also possess flagella, but these are structurally different from that. They want to say that, yes, guys, flagella also present in eukaryotes. Eukaryotic cell also contain flagella, but they are different with prokaryotic. The electron microscope study of cilium or flagellum show that they cover the plasma membrane. They covered what? Look at here. They covered plasma membrane. They covered plasma membrane. Their core called axoneme possess a number of microtubules running parallel to long axis, and axoneme usually have 9 doublet and radial arranged peripheral. They want to say this thing. I told you now, remember this structure, you have clear everything. What is this? This is the peripheral tubule. Some are central microtubules. Okay. They want to say that this is they are present in 9+2 arrangement. Look at here. The central tubules are connected by bridge and also enclosed by central sheet. The central tubules are protected by, these are microtubules, they are protected by central sheet. Guys, this is the number of microtubules. 1, 2, 3, 4, 5, 6, 7. They may be 12 to 13, guys. They may be 12 to 13 in number, minimum, and maximum according to vary in size of eukaryotic cell and prokaryotic cells. Means we know, guys, this is the fixed number. The 9 are fixed in number, but the microtubules amount between them, that are not fixed. It may be 12 in number, it may be 12 in number, it may be 13, 14, 15, 16, 20 number. Okay, guys, now clear? Now this thing is clear? Okay, next is, okay, there's all. There are 9 radial spokes, okay, we know this thing. The peripheral doublets are also interconnected by linkers. Both the cilia and flagella emerge from centriole-like structure, call it as a basal body. Now our next organelle is centriole and centrosome. In cell division, we already know what is centrioles, how they look. I'm drawing like this manner. They divide in S phase, G1 phase, G2 phase. Centrosome is the organelle usually containing two cylindrical structure called centriole. Two cylindrical structure who contain centrosome. They are surrounded by a amorphous pericentriolar material. They are covered with this kind of material. Both the centriole in a centrosome lie perpendicular to each other. Look at here, I am drawing one centriole and other is perpendicular. Clear or not? They are made up of 9 evenly spaced peripheral fibrils of tubulin protein. Each of peripheral tubule is a triplet. Now this is the 9+2 arrangement. But they are, what is the difference in centriole? They are, I'm doing here, they are triplet in number. Like this arrangement, triplet in number. Alright or not? Okay, 3 set. They are 9 guys, 9. Of course they are 9. 1, 2, 3, 4, 5, 6, 7, 8, 9. They are 9 in number. They are 9 in number, but triplet set. Now clear guys? Now clear? Okay, the central part of the proximal region, proximal means near region of centriole is also proteinaceous and called the hub. The center part we call it as a hub, which is connected with tubular of the peripheral triplets called radial spoke. Look at here guys, this is the radial spoke. They are connected with central sheet. This is the radial spoke. Same structure guys, same structure. Okay, the centriole form the basal body of cilia guys. Here, this is the base part of the cilia and this is cilia rise. They are useful in movement and making spindle fibers. You know now, here is centriole and there is what? Spindle fiber. And they attach with centromere. And kinetochore. This is the chromosome. This is the centromere. This is the kinetochore. Spindle fiber attached with this. And they are connected with centriole guys. Alright. Now let's start with nucleus. Guys, nucleus is the structural and functional unit of the cell. This is the cell, and the center part. They contain genetic material. We call it as a nucleus. Described by Robert Brown in 1831. The material of the nucleus stained by basic dye was given the name chromatin. When Fleming, this is the scientist, when Fleming watched nucleus, they found some chromatin, thread-like structure. They saw a thread-like structure. They call them chromatin. Where chromatin is located? Chromatin located in nucleus guys. The interphase nucleus has highly extended, elaborate nucleoprotein fiber. We call it as a chromatin. This is the definition of chromatin. This is the long, extended, elaborate nucleoprotein fiber-like structure. We call it as a chromatin. This is the definition. Nuclear matrix and one or more spherical bodies called nuclei. In nucleus, they also contain a dark region. We call it as a nucleolus. Okay, now when scientists watch in electron microscope, what they saw? The nucleus have their nuclear envelope, which consists of two parallel membrane with a space between called perinuclear space. They call what they say that nucleus covered by nuclear membrane, and this membrane we call it as a nuclear envelope. This is the pore guys. Nuclear envelope and space between what? Space between outer layer and inner layer. We call it as a perinuclear space. This cell space filled with fluid, and this is necessary. So that's why both the membrane can't overlap each other. Okay, forms a barrier between the material present inside the nucleus and that of the cytoplasm. Means this is the cell, and this is the envelope. This is the nuclear envelope. And what they prevent? So outer cytoplasm can't come inside and inner nucleoplasm can't go outside without their permission. But we know now at the nucleus ER will attach, so they can help with their pore. They transfer their message in ER, and ER transport their message Golgi body, and Golgi body transport their message in the form of vesicles. We call it as a vesicle, and they transport their message towards the lysosome, and lysosome transport their message towards the plasma membrane. We call it as an endomembrane system. The outer membrane usually remaining continuous with ER. Now clear guys? Now you clear that this outer part is covered with and ER attached with the outer surface of ER. If Endoplasmic reticulum bears carry ribosome, we call it as a rough ER. If it doesn't contain ribosome, we call it as a smooth ER. At a number of places, the nuclear envelope is interrupted by minute pore. Here, these are the pores, which are formed by fusion of its two membrane. When these two membrane fuse, the pore between present in this membrane. These nuclear pore are the passage through the movement of RNA. We know now here DNA is present. DNA form in future RNA. RNA goes out with the help of this pore. Any kind of message nucleus can transport with the help of these pores. Okay. Protein molecules takes place in both directions between the nucleus and cytoplasm. Normally there is only one nucleus per cell. Variation in number of two of nuclei are frequently observed. What they want to say that we know that RNA come out with the help of these pore. Any kind of signal message come out with this pore, but they require protein. What guys? They have protein on their membrane inside or outside. They transport these molecules with the help of pore. Means this is the protein part, a special kind of protein. They collect message and they transport with the help of the pore outside the membrane, and this message cross and this protein again return towards this inside. Clear? If they transfer any kind of signal, what they do? They have protein, they take any kind of message signal and they come out with the help of pores, and after dropping outside this signal they goes inside. So the name of the protein who throw signal outside the nucleus, we call it as a exportin. They export any kind of RNA, any kind of protein. But if any protein want to come inside, then these protein take this kind of protein and they enter with this pore and give the protein and again go outside. If something is importing inside, we call it as a importin, and this is the exportin. This is the membrane, this is the pore. If someone want to go outside, they bind with exportin come out and give message to another cell organelle. And some signaling want to come inside, they bind with importin and they import inside the nucleus. So that's all for nucleus guys. They have genetic material guys. So let's study genetic material. We call it as a chromosome. If I say this is the thread like structure, we call it as a chromatin. If chromatin plus histone supercoil, this is the stone octamer, this is the stone octamer, this is the stone octamer, and DNA wrap around. If they compact, they make chromatid chromosome. This is the chromatid. The center is centromere. Okay, they condense, and what they do? They make like this structure. Now clear? Now clear guys. And they look like this in future. Here is condensing. Now you can better understand. They condense multiple times. They condense. Okay, this is the centromere and kinetochore. Cover this centromere. These are the kinetochore. Okay, now according to centromere arrangement, the chromosome divided into metacentric part, submetacentric, and acrocentric. Look at here. If centromere present in the center part, these are the what? Centromere. They present in the center part. This size equal. We call it as a metacentric. If they present unequally, top side of the chromosome, this arm is unequal, this arm is unequal, we call it as a acrocentric. If this centromere at the top of the chromosome, look at here, now this, look at this, we call it as a telocentric. Look at here, the submetacentric chromosome has a centromere slightly away from the middle of the chromosome, slightly away from the, these are the submetacentric. And metacentric is the chromosome has middle centromere. This is the middle centromere, this is slightly away from the middle, we call it as a submetacentric. And the acrocentric chromosome, the centromere is situated close to the end, forming one extremely, look at here, and they present in the ending part of the chromosome, terminal part of the chromosome. So this is all about chromosome. And now the last is microbodies. What is microbodies? Many membrane bound minute vesicle, call it as a microbodies, that contain what? Of course enzyme, and they are present in both plant as well as animal. So here your chapter is also complete. So we will meet in our next video. So thank you guys, thank you for listening me, and please subscribe my channel, keep revising. Okay, thank you.